Hello students, welcome back again in my class. In the previous class we have discussed our first topic, that is nucleic acids and their classifications. We have discussed about what are the nucleic acids and what type of nucleic acids we can have and their various fragments. In this particular lecture, that is topic 2, that is nucleosides and nucleotides, we'll discuss about these two highly important topics or terms. If we just a second, if we look about this nucleosides, basically, these are small molecules and they are formed when the pyrimidine or purine base combines with a five-member sugar that can be your ribose or 2-deoxyribose sugar, and a nucleoside is formed. ठीक है? For example, if we combine adenine, that is a base, it is a adenine, this is your purine based base, and we combine this with ribosomes, so they form a nucleoside. Okay, we'll get a nucleoside, and that nucleoside will be known as adenosine. You can see here in this structure. So this is our base, that is adenine, and this is connected to this ribose sugar by at this bond, at this carbon through this bond. That is, this bond is known as glycosidic bond, or it's called beta glycosidic bond. We get it. Okay, so the base and this sugar, they are connected with other with this beta glycosidic linkage at this carbon. That is one dash carbon. Is one carbon of this sugar, and the formed molecule is known as nucleoside, and the name is adenosine. The same way, if we combine guanine with, let's say, 2-deoxyribose, we'll get another type of nucleoside, that is deoxyguanosine. Similarly, if we cytosine combines with ribose, the formed nucleoside is known as cytidine. When uracil combines with ribose, we get uridine. When thymine combines with the deoxyribose, we get thymidine. Now, here I have one question for you that will appear on your screen, and you have to answer for that. Now, these nucleosides which contains ribose, they are termed as ribonucleosides, or which contains ribose sugar. Actually, they're known as ribonucleosides, and those which contains this deoxyribose as a sugar moiety in their structure, they known as what? Deoxyribonucleosides. Okay, then depending upon the structure of sugar we have in the nucleoside, we can name them as ribonucleosides or deoxyribonucleosides. Okay, now they are, you can say, glycoxylime obtained by chemical. These actually glycoxylimes, they obtained by chemical or enzymatic decomposition of nucleic acids. Means when an enzyme or a chemical attacks on these nucleic acids, they break their structure and we obtain these nucleosides. Okay, in a nucleoside, a base form that glycosidic linkage with sugar and are of non-reducing nature. Just I told you that there is a bond between base and sugar at C1 carbon, and because of this bond at C1 carbon, now this carbon is not free. If you look at the structure of free structure of these sugars, they contain aldehyde group, and aldehyde groups, they are of reducing nature. They can reduce the tolerance reagent or filling reagent. Okay, you must have studied all these tests. But in this case, where we talk about nucleosides, they are non-reducing nature. That means their C1 carbon is blocked. They don't have a free formyl group or free aldehyde group, and which suggests or confirms that the linkage between the sugar and the base happens at C1 carbon of the sugar, and because of that, they show non-reducing nature. Okay, and this bond is known as beta glycosidic bond or beta glycosidic linkage. Now, these are the structure of that cytidine and thymidine and uridine, that nucleosides formed by the combination of cytosine, ribose, thymine, 2-deoxyribose, and uracil ribose. Again, I have a question here for you guys. Just that will again appear on your screen. You have to answer for that. Now, coming to the nucleotides. Nucleotides, the other part of, you can say, through a little bit bigger part of nucleic acids other than the nucleosides. If you look the structure of this is a structure of nucleotide, and you have seen previously the structure of nucleoside. Can you distinguish between these two structures? Can you differentiate? Just try once. Okay. I am again giving you a question. Just try that question. Whether let's see whether you can distinguish between these two or not. Okay. So when a phosphoric acid, H3PO4, it's acidified to one of the hydroxy group of sugar portion. Okay, sugar portion of a nucleoside, we obtain a nucleotide. That means the difference between a nucleoside and a nucleotide is that in nucleotide we have a phosphate group in addition to the nucleoside. Okay, so therefore this nucleotide contains a purine or pyrimidine base, ribose or 2-deoxyribose sugar, and a phosphoric acid unit you can see here. This extra part you can see in nucleotide. Okay, now the attachment of this phosphoric unit to the sugars, it can be at any carbon which contains a hydroxy group. For example, in case of ribose, we have hydroxy group at 2 dash position, 3 dash position, and 5 dash position. So this position is not 1 dash, this is 2 dash, this is 3 dash, this is 4 dash, this is 5 dash. Now 4 dash पे हमारे पास कोई भी OH group नहीं है. We don't have any OH group at this carbon. We have here one OH group. If you look at the structure of nucleoside. Okay, so at these position, either at this position, that can be obtained. I'm talking about the ribose. Okay, in 2-deoxyribose, we have hydroxy group at, you can see, at 3 days and 5 days. There's no hydroxy group at 2 days. Okay, this is the difference between ribose and 2-deoxyribose. So this phosphoric acid can be attached at 5 days, 3 days in 2-deoxyribose, whereas in case of ribose, it can be at 2 days, 3 days, or 5 days. Okay, the position can be any. Okay, now naming of these nucleotides. Okay, so a nucleotide is named for the parent nucleotide with the suffix monophosphate or diphosphate, depending upon the number of phosphate units added to the sugar system. So whether we have one phosphate group or two phosphate group or three phosphate group attached to the sugar system, we can name the nucleotides as monophosphate, diphosphate, triphosphate. Okay, depending upon the number of phosphate groups attached to it. For example, in adenosine, we have seen the structure of adenosine in the previous slide. So where we have seen there is one phosphate group attached to this 5' carbon. So this can be named as adenosine 5' monophosphate. This 5' represent the position of phosphate group attached to the system. Likewise, if we have two phosphate group here in this structure, you can see here there is one phosphate group, there is another phosphate group. So this type of system can be named as adenosine 5' diphosphate. The common name is ADP. You must have heard about ADP and ATP. ATP is triphosphate system. You can see here. This is the triphosphate system. Adenosine 5' triphosphate. Okay, ATP. Now you may have a question in your mind, sir. All the phosphate groups are attached with each other in a, you can say, linear chain. Whether they can be present in either positions, yes. In this diphosphate system, the one phosphate group can be here, the other phosphate group can be at this carbon, that is 3' carbon. And in that case, we have to assign both the positions. We can name it like adenosine 3' comma 5' diphosphate if we have phosphate group at 30' position and 5' position. Likewise, if we have a hydroxy group at, let's say, 2 dash, 3 dash, and 5 dash, we have to name it like adenosine 2 dash, 3 dash, 5 dash diphosphate. So we have to specify the position of the phosphate group present in this nucleotide system, as well as the number of phosphate by assigning mono, di, tri, tetra, etc. So I hope you have understood about the basic concept of nucleosides and nucleotides. Okay, and I hope you'll be able to attempt the questions based on these. Okay, let's see how much you have learned. I'm giving just a small task to do. You have to do that, and we'll see how much you have learned in this. Thank you so much.